Welcome to the honors lecture on DNA replication. We're going to talk about how new DNA is created during the S stage of the cell cycle. So before we move on, I need to make sure that you understand these concepts here. So in the Edpuzzle, please go ahead and type in your answer regarding these questions here. I am going to pause it while you do that, so please make sure that you type in your answer. Our goal for DNA replication is to replicate DNA, which means to make a copy of something. So this is semi-conservative, meaning that the DNA strand is half old and half new. So some of these things about this process here. Again, I'm going to have you type in your answers into the Edpuzzle here. So please make sure and type those in right now before we move on. So some things about DNA make it really complicated to copy. First of all, DNA is polar. So what does that mean? It means that it is a charged strand. So one end has one charge and the other end has another charge. However, because it is double-stranded, both sides have both charges. And in that way, we see that the sugar side of the DNA has a positive charge, and we call that the 3' side. The phosphate side has a negative charge, and we call that the 5' side. So the way that the enzymes work in building the strand is that they move in the 3' to 5' direction. But that means that they build the new strand in 5' to 3'. So let me show you what I mean with that. Here is our DNA strand, right? So because it's two-stranded, this strand here is polar, and this strand here is also polar. Basically, one side is just flipped over. That's why the P's on this side are upside down. So in order for the DNA strands to stick together, they have opposite charges. So this 5' side with the phosphate strand over here is negative, and this 3' side with the sugar is positive. So the negative and the positive help to hold these two strands together. The hydrogen bonds in the center are not strong enough to do that on their own. So it's important to note that the other way that DNA sticks together is by the charges of the strands here. So if this side is negative, then down here this side is positive, and that means this side is negative and this side is positive here. So the enzymes can only move in the 3' to 5' direction, which means that they're going to move along a strand this way, meaning that they have to build the other strand, the other one that's shown here, from the 5' to 3' direction. So people that came up with this were Meselson and Stahl. They did several experiments to determine that DNA is replicated conservatively, dispersive, or semi-conservative. It turns out that it's semi-conservative. Conservative would mean that one strand is completely old and one strand is completely new. Turns out that's not what happens. Dispersive would mean that it's all mixed up. That's not what happens. Semi-conservative means that one strand is old and one strand is new. So here are the steps that Meselson and Stahl determined were making the copy of the DNA. So first thing that happens is that we unwind the DNA. This is when the helicase breaks the hydrogen bonds so that the helicase, the next enzyme, can get in there to add them. Step number 2 is that they are stabilized by the spooling proteins. Now in some places you'll see these called different names. They're the same as the histones that the DNA wraps around when it's not being duplicated. And they stabilize the open strands because they want to stick together. Remember, they're opposite charges, so they try and get back together. The histones prevent them from doing that and making sure that they aren't breaking. Next, we take a copy of the DNA by adding complementary nucleotides. That's the job of the DNA polymerase. If there's an A on the parent strand, it'll add a T, or if there's adenine, it'll add thymine. So they add them in a particular order, and they have to go only in one direction. So after those have been added, then we check for errors. Ligase is the enzyme that does that. It also connects things together, and polymerase also checks for errors. And lastly, the fragments get connected together. So we have little fragments on the ligang strand, which I'll show you in just a moment, and those are connected together by ligase. Right, so because of this semi-conservative replication, we have one strand that's completely old, and then we add nucleotides to that strand to make a second side, which is completely new. So because of the charges on the DNA, the charges cause the polymerase to move only in one direction. And because the two strands are opposite directions, that causes the polymerase to move opposite on those strands. Okay, so we come up with a leading and a lagging strand. So that causes some issues here. So as the helicase unwinds the DNA, on the leading strand, the polymerase can just follow the helicase. It just follows it and creates one long strand of DNA. But on the other side, the polymerase has to move away from the helicase, since it can only move in that 3 to 5' direction. So that creates a bunch of little segments on the DNA that we call Okazaki fragments. And those Okazaki fragments need to be connected together. So the replication enzymes move along the original DNA from 3' to 5'. So we can't build the new strand 3 to 5 because then we would have two positives next to each other and two negatives next to each other. So as we move from 3 to 5, it builds the new strand in the 5 to 3' direction. So the parental strand, the original strand of DNA, the side that starts with 3' is the leading strand, because then the polymerase can just follow the helicase as it goes down. The one that starts with the 5' is the lagging strand, because the DNA polymerase has to move away from the helicase and then loop back and add more nucleotides that way. So here's a diagram to show that. This is the leading strand. Helicase is moving in this direction, unwinding the DNA. The place where it's unwinding is called a replication fork, so it moves in that direction there. On the leading strand, which is the 3' parent strand, we can build the new one from 5 to 3 and just follow the helicase. On the lagging strand, however, since the parent strand is the 5' strand, we have to build away from the helicase. So the polymerase would move away from the helicase here, and then loop back and move away here, and then loop back and move away here, creating all of these little fragments called Okazaki fragments. The ligase will then come in and fill in these little gaps and connect everything together while it proofreads. Here's another diagram of the same process. Here is our helicase unwinding the DNA. Here are those histones, also called the SSB or single-strand binding proteins. They stabilize the DNA. Here's our DNA polymerase, and since this is the 3' parent strand, it can just follow it completely, building the new strand in the 5' to 3' direction, while the moves along the parent strand from 3 to 5. Here, we have the 5' parent strand. So the ligase can't move in toward the helicase. It has to move away from the helicase. So it would start by moving out over here, and then loop back, and move out away, and then loop back, and move out away, and loop back. And as the helicase continues to unwind, this is going to continue to move in that direction. And then we have a ligase that's going to come in and fill in all of these gaps and connect everything together so that we have two complete strands of DNA. Okay. Please go ahead and scroll down so that you can see some more videos that describe this process, and don't forget to answer or send any questions to your teacher as soon as if there's anything that you don't understand.